In this second scientific method video, I want to go through the scientific method now using a biological example. So in the previous video, I showed you the steps of the scientific method. Remember, those steps are to observe, question, generate a hypothesis, experiment, draw conclusions, and then report and publish. What we're now going to do is talk about how we would use the scientific method based on sort of a biological observation. So let's say, as a scientist, you were out and about and you noticed in different fields and fields that were maybe right next to each other, that the plants were significantly different from one another. Plants in field A, let's say, appeared to be more healthy. They had more leaves, potentially they produced more fruits. The field right next to it, right, field B, the plants were significantly smaller, less leaf growth, less fruits. And let's assume that the plants were exactly the same. Maybe they're tomato plants, for example, right? So that's an observation that you made. So the next step, right, after making that observation is to develop a question. So the question might be, well, why are the plants grown in field A so much healthier, quote unquote? Why do they have more leaves? Why do they have more fruits than those grown in field B? So once you've developed your scientific question, right, the next thing we need to do is to generate a hypothesis. So based on your background knowledge, maybe you suspect that the soil content, that fertilizer was placed on fields. Perhaps field A has more nitrogen. Maybe it was fertilized more than what you're seeing in field B. So maybe the explanation, the hypothesis that we're testing here, is that field A has more nitrogen in the soil than field B. And that's leading to the difference that we're seeing in regards to the plant growth in regards to leaves and fruits. Now remember that a good hypothesis should be testable, which of course we can test this, right? It should be falsifiable, meaning, you know what, we might find that nitrogen has nothing to do with it, right? We won't know until we test it. And then finally, it needs to lead to predictions. So our prediction here is, well, if nitrogen leads to more leaf growth, more fruit production, then fields that have more nitrogen should have more leaves than fields without nitrogen. So again, it's sort of that if-then statement, right? If the hypothesis is correct, then this is sort of what we expect to see in regards to our data. So the next thing that we need to do is we are going to propose an experiment. We have to come up with an experiment that's going to test our hypothesis and provide data that's either going to support the hypothesis or it's going to fail to support our hypothesis. So when we generate or set up our experiment, we have to take into account some variables. All right, and so there are three types of variables we're going to talk about. We're going to talk about independent, dependent, and standardized variables. So it's very important that not only students know the definition of those terms, but given a scientific study like what we're doing right now, that you could identify what is your independent variable in a scientific study, what is the dependent variable in a scientific study, and what are these things that are called standardized variables. Well, let's start first with the definitions. So an independent variable is basically what the researcher, it's what we want to test or know the effect of. It's really the variable that we are controlling. So what are we controlling here? Well, we're controlling the amount of nitrogen that's being placed on the fields, right? That's what we're testing. We think nitrogen is really affecting plant growth. And so when we're doing this controlled experiment, what we're interested in testing, what we really want to know the effect of, what we're altering, is the amount of nitrogen or fertilizer that's potentially put on these different fields. Now, the dependent variable is what is being affected in the experiment. This is what we think is going to change when we manipulate the independent variable, okay? This is what we're going to measure or we're going to observe. So if you think about this, right, if we look at our two little these are two fields, right? What are we observing? Well, we're observing the number of leaves, right, on the plant, excuse me, and we're observing the number of fruits that the plants put out. So again, the dependent variable here is what we're going to measure. It's what we're going to observe to see ultimately how the independent variable is sort of affecting, okay, the study. So the independent and the dependent variable are really the first two variables that we really want students to be able to sort of, when we're looking at a scientific study, be able to identify those. Now, standardized variables are variables that may affect the outcome of the experiment, and therefore, as a result, we have to keep them the same. Okay, we have to keep them the same between sort of our two groups here. So what are things that affect plant growth? Okay, well, we know that plants need light energy, and we also know that plants need water. So those are two examples of standardized variables, right? We have to make sure that both sets of plants receive the same amount of light and water, because we know, right, if we water one set but not the other, then that could impact our results, right? And so the differences that we see might not be because of the independent variable. They might not be because of the nitrogen, the fertilizer, but instead it can be because we didn't control for these sort of standardized variables, and that impacted our results. Now, when you're setting up a controlled experiment, usually what we do is we're going to break sort of the experiment into sort of two separate groups. We're going to have what's called a control group, and we're going to have an experimental group. Now, the control group is the group where the independent variable is either absent or it's set to a predetermined baseline value. So, for example, there are cases where you may not be able to completely remove the independent variable. So let's say that you hypothesize that in eukaryotic animal cells that oxygen levels impact digestion. Okay, I'm just kind of making this up. But one of the things to recognize there is, okay, well, we want to look at the difference of oxygen consumption between different animal cells, right? And what we want to see is, how does that affect digestion? We think the more oxygen, greater levels of digestion. Well, eukaryotic animal cells, they have to have oxygen to generate ATP. So we can't completely remove oxygen or it will end up killing the cells. So in this example, the independent variable, right, in this case, the amount of oxygen given to the cells, there has to be some sort of predetermined baseline value. And then on what we would do in the second group, our experimental group, is we would increase the level of oxygen that we would give the plant. Again, we would vary the independent variable. Now for our example, right, it can be a little bit more straightforward. If we're testing, nitrogen in the form of, maybe fertilizer that we're providing for the plants, you can see here, right, we won't fertilize this series of plants. This would be our control group. This is where the independent variable is absent. And then we have our experimental group. That's the group that's subject to a change in the independent variable. This is where we're going to apply the nitrogen, right, in the form of fertilizer. Now, a couple of other things that we have to think about with our experiment. The first is the sample size. So sample size is going to represent the number of individuals or samples being tested. So we want to make sure that we have a large sample size so that we feel that the research that we're conducting and that the results that we get are unbiased, that the data that we're obtaining is significant. If you only have one tomato plant and one tomato plant, again, with such a small sample size, the data is very unreliable. So a big, large, robust sample size is very important when you're carrying out these experiments. In addition to that, we want to make sure that we repeat the experiment several times. Usually scientists like to repeat an experiment at least 3 times to verify that the results are consistent. So when we talk about replicates or repeating the experiment, again, that's important because again, it it verifies that what you're seeing you didn't just see it once, that this is something that's consistent and significant. So keeping that in mind, of having a nice big robust sample size and also repeating the experiment, these are all things that you need to think about as you're designing your experiments. Now, once you design your experiment and you run your experiment, you're going to collect data. And when we collect our data, we usually present that data in both tables and graphs. Tables are used to summarize and present the results in their actual numbers. So you can actually see that there's numbers here in regards to how much nitrogen you're putting on, what the plant growth was, right? In regards to whether it's leaves or fruits, right? This is a table I quickly just kind of threw together. But there's a couple of things that when you're looking at the table should sort of, pop out at you. When you generate a table always sort of has a name and a number. So this is table, 1.1. In addition to that, there's always a description. The description basically tells you the contents of the table. It should be concise. It should, include the key elements shown in the table, right? So this table shows the effect of fertilizer on plant growth. When it comes to graphs, okay, they provide a great visual way to show your data, right? They show the relationship between the independent and the dependent variable. They really highlight that important data. And if it's generated correctly, it's usually self-explanatory, right? So we can quickly look at this and we can see by looking at the fact that as we increase the concentration of nitrogen, right, we see more plant growth. Now, one of the things to keep in mind is that when we are generating our graph, the independent variable gets graphed on the x-axis and the dependent variable gets graphed on the y-axis, okay? One of the things that I also want you to recognize when you're doing graphs is that there are some important components that always need to go with a graph, just like with a table, right? There should be a number, right? There should be a brief description that tells the contents of the figure, right? It should be concise. It should be clear. Again, the y-axis that you see here should always be the dependent variable, and the x-axis represents the independent variable. Always make sure you include your units when it comes to graphing. Okay, so several sort of key components there when you're generating tables and graphs, and you will be asked to do this throughout the course. Now, the last two steps to the scientific method are drawing conclusions and then reporting and publishing. So at the end of the experiment, you're going to look at the results, okay, and you're going to draw some conclusions. The results are going to be used to help you determine whether or not the hypothesis was supported or whether or not you failed to support the hypothesis. Remember, it doesn't matter if the data if the data doesn't support your hypothesis. That's still important information. But it's very important when you generate those final conclusions that you do circle back and address your hypothesis, okay? Once you have drawn your general conclusions, the next step that we want to do is to report and publish. So once the experiment's complete, results analyzed, right, conclusions drawn, typically what we do is we publish our scientific work, and scientific work is often published in scientific articles. Articles are typically published in many different ways. They can be published on a non-reviewed site on the internet, or they can be published in what we call peer-reviewed journals. Peer-reviewed journals are journals that contain papers that have been rigorously looked at by sort of experts in the scientific community in that field before they're published. So not just anybody can publish in a peer-reviewed journal. When you submit knowing that your work is going to be sent out to experts and be reviewed. And when they review that, they're going to look to see how well did you set up the experiments. Does your data really support what you're saying? If they find that the study is solid, the article will be published and you'll get this nice accept. If they feel that there are holes in the article or there's holes in your experiment, then what they often do is they will not accept the work and they will give you a lot of feedback on how you can go back and reevaluate, and then you can resubmit. it's very, very important that results get published, because again, it's a way for us to share knowledge within the community, the scientific community. It is also important that students recognize that not everything you read is peer reviewed. So sometimes authors are just publishing things on the internet or wherever, and they haven't really been confirmed by anyone. So it is important to understand sort of these different ways that scientific work can be published. So those are the steps of the scientific method. I've gone through them now with you in regards to using both a general example and a scientific example. Now, the last thing that we typically do in this lab is we have students sort of design their own experiment to work through the scientific method. Now, if students are doing this at home, that might be that your instructor gives you a set of instructions, something that you observe, and then you generate questions and hypotheses, and you're able to carry out that experiment. It might be some sort of a virtual experiment, okay, that we do. Or again, if we are able to meet in lab, oftentimes the experiment that we have students work through is really this, what we call sort of an acid experiment. Antacids are a type of medicine that can be used to raise pH. So your stomach tends to be a very low pH, okay, very, very acidic, and acids should bring the pH up. It should cause the pH to go up. There's a little example in your lab book on page 9, where it talks and it kind of gives you a background about antacids. When students are in class, they're basically working through how antacids work based on surface area. They're asked to generate questions, design experiments. We give the materials to do this. Again, depending on whether we are meeting in the lab or whether students are carrying out the lab at home, you may be asked to do something a little different depending, again, on the situation. But with that, I want to finish this video, and again, I want to make sure that students understand that instructors are there to help you navigate through this. And so if there's any confusion or if there are any questions, please don't hesitate to make sure that you reach out to your instructor.